So let's go ahead and get started today. It's a real honor and privilege to introduce our speaker at this Hanover seminar, Professor John Rao from the University of Wisconsin, Department of Biochemistry. He's also, as a leadership role in the Great Lakes Bioenergy Research Center, he received his bachelor's degree in chemistry from Canterbury University in New Zealand, which is where he's from, and a PhD in forestry and chemistry from the University of Wisconsin. So I looked at Clarivate Analytics today, and they've got him ranked as one of the top 1% researchers in his field. He's been cited over 38,000 times and has an age index of over 103. I was in the forestry, Beijing Forestry University in 2013, and his picture is on the wall outside of Roon Kang's students' lab, who does a lot of work in Lignins. So it gives you an idea as to how well known he is. So with that, I think he's one of the preeminent researchers in lignin chemistry in the world. And we just lost our tech support.
Okay, forget that idea. Keep this quicker.
What happened? Yeah. We're back. Okay.
Okay, we're not touching
Professor John Rao.
All right, so someone let me know when 3 hours is up, all right? Okay, I'm flustered. Thanks, it's a pleasure to be here. I wish I didn't go through that nonsense, but never mind. I don't know if I targeted this right, but I'll try and tell you something new about lignins that you might not have known about, and talk about how we're manipulating it in plants for our own use. Okay, little introduction. Lignocellulosic. The term is lignocellulosic. It comes from cellulose. I've been on a campaign to try and stop people, to have people call it cellulosic and stop calling it cellulosic. I don't know if people do that here, but I wish they would stop. And of course, we had a particularly eloquent member of the previous administration who called it cellulosic. Let's not do that. Okay. So the problem, what's the problem? These cell walls are the problem. They're gorgeous structures. Some of us like these things a lot. The cell wall is mostly sugars, made out of mostly sugars. I'm not going to go into all this stuff because I think you've seen this before. But they weren't designed for us to get at these sugars, right? They were designed for the plant. Compositionally, half of it's cellulose, a third basically is lignin, and a third is hemicelluloses or whatever. Grass is the same kind of thing. All right. What would be nice is if we could fractionate the components first, because pure components we can deal with much more easily than the big mixture. So get the cellulose out, the hemicelluloses, and get the lignins out. And this usually means getting the lignins away from the polysaccharides. The problem with that is that getting that lignin out is very energy demanding. It takes something like one of these things, a pulp mill, to do it. All right. Often you'll see lignin is only good for this. That has value. So anyone denigrating lignin as having no value because they just burn it, that burning has value actually. But it has value beyond that. We're starting to realize that. It's got high fuel value, and it's one of the largest sources of aromatic components on the planet, sustainable aromatics. All right. A little bit about initial thoughts on dealing with this lignin problem, the problem of trying to get lignin away from the polysaccharides. We've understood that there's a pathway, a synthetic pathway to make lignin. I'll tell you what lignin is a little bit in a bit, but not enough. But basically we have a pathway. It's not supposed to be a dog, by the way. This is the pathway to the 2 monolignols. And of course, given that we know the pathway, why don't we just knock this stupid thing out? Can't we just get rid of that if lignin's a problem? Well, part of the problem is sort of that, but the real problem is sort of that. Plants really need the lignin. They've evolved to use it for strength and water conduct and all the rest. So you can't just get rid of the problem. But you can get a fair bit of information, a bit of insight into what kind of things you can do by perturbing lignification. Anytime you can perturb a system, you get a nice way of understanding something about the system. In the past, we've really only had one way of perturbing it, the natural way of somehow perturbing the plant system. Some of these ways are distinctly more natural than others. I don't know who decided on that experiment. But now, of course, we've got this way using these really ugly genetic engineers to poke and prod at individual genes on the pathway. And so there's a lot of plant materials around now where individual genes for enzymes on the pathway have been poked and prodded to see what happens. And basically there's now a whole set of poplars, for example, including these bright red ones, transgenics with each of those genes missing in the pathway. All right, that's all I'll say about that. A couple of things about lignins. First of all, I'm not going to go into this in big detail, but the lignin is formed from these 3 hydroxycinnamyl alcohols. They just differ in the degree of methoxylation. So coniferyl alcohol and synapyl alcohol, and these make these G and S units that I might inadvertently use. They're made in the cytoplasm, and they're exported somehow to the wall. You can make a model for the lignin. It's only a model. Doesn't mean very much. I'll come back to it, but I have a point about this in a second. And you can do modeling on it. Big, hairy deal. A couple of things about the lignin polymer. First of all, this process of lignification, it's about radical coupling. Some of you are good chemists and you've done 2-electron mechanisms. Some of you are weaker at single electron mechanisms. I'll just show you the way this basically happens and make a real important point about it in a second. Basically, you make a phenolic radical, and you make a radical of the growing polymer. So this is the phenolic end of the growing polymer. And then you just simply push electrons, and you take 1 electron from each of these species, and you make a new bond with 2 electrons in it, 1 electron contributed from each of those species. So in this particular case, you make this beta O4 bond, for example. It's the most common one in the polymer. By pushing electrons just a slightly different way, you can make beta 5 coupling too, and if you don't know chemistry, this makes absolutely no sense, but this is how you get this kind of combinatorial coupling of radicals to make various products from the same original starting materials. Here's the important point. When you do this coupling, so beta 4 coupling for example, you make this intermediate, it's a quinone with I, you make this new beta O4 bond, but you also make a new chiral carbon. A new carbon that has a different left and right hand from it. You make that thing chiral, but the product is not optically active, and that tells you something. You re-aromatize that intermediate by adding water to this thing, and when you do that, you make an OH here, and you make another chiral carbon. So you make 2 chiral carbons for each time you do this coupling and re-aromatization. Again, that product is not optically active. The ramification of that, the implication of that, is that this process in the plant is purely chemical. We're used to processes in the plants being dictated by, controlled by enzymes and proteins that couple things in very specific ways to make a very specific molecule. Lignin is not like that. It has some implications. First of all, lignin's a racemic polymer. Doesn't mean much when you just say it like that, but here's the implication of racemic nature. Okay, thank you. Yeah. So if you make a model at 20 mer, again, just for illustrative purposes, looks like this. So you have 20 units, you just made 19 couplings, you've got 38 optical centers in that thing. 38 optical centers. That gives you 2 to the 38, if you remember your first year of chemistry, optical isomers. Half that number real isomers. Some of them are fixed relative to the others because they form these ring structures. Don't worry about it. But in fact, then you actually have 2 to the 35 optical isomers. 2 to the 34, half that number, real physically different compounds. Siri, what's 2 to the power of 34?
Here's what I found. Siri was 10 to the power of 34.
Yeah, well, she was supposed to tell us, wasn't she? It happens to be, anyone else do it in their head? 17 billion. 17 billion isomers of that one structure. I'm not saying they're all formed, but there's that many possibilities. So when someone tells you, I've just made a model of lignin and I've minimized it on the computer, ask them which of the billions of isomers they made and whether it matters. This is underappreciated. All of these possibilities, physically distinct possibilities, are in the lignin polymer itself. So there will be no 2 molecules in a plant that are the same. Very different than your other polymers. Your proteins are all made from L amino acids, yeah, amino acids, right? All linked together and you form one kind of molecule. Every single molecule in the plant is actually different because of this isomeric complexity plus the fact that they get pieced together in a different order as well. All right, so this makes for complex, for example, in a mass spectra. If you take a little dimer, the peaks are very nice. You get to see these little alpha, gamma peaks for a dimer. You just have to make a trimer, and we were able to make it in such a way that you make us 4 of the 8 isomers and the other 4 of the 8. And I think you can see that if you put these things together and you broadened it out a little bit, you've got a freaking broad mess. And that's what lignin looks like. All the sharp peaks in lignin are not real peaks to do with the polymer. These broad, fat things here, those are the peaks in lignin. And the reason they're that broad is because you're just looking at no two molecules the same. Every molecule is different. So then they all have slightly different chemical shells. Compare that with a higher molecular weight protein, and every peak in that protein is actually sharp. It's still a mess, but each peak is sharp. It's nothing like what the lignin is. All right. That's one. So lignin, it is complex. There's billions of isomers, billions of structures possible. Compositionally, there's also lots of authentic monomers, and I'll hopefully remember to point out why that's the case in a second. And so structurally, it's just really, really complex. But the process of making the polymer, lignification, the polymerization process itself is dead simple because it's just one reaction. It's just radical coupling chemistry. It can go various ways, but it's just one simple reaction. There's no enzymes or proteins involved in the polymerization, and because it's purely chemical, that means any phenolic compound you can send to the cell wall can be incorporated into the polymer just subject to its chemical compatibility. It doesn't have to have an enzyme to connect it together. It just has to be chemically compatible. And so you can actually make lignins with other components in it, which is something you can't normally envision doing with other polymers. All right, so we want to engineer lignins. Why? Would be nice if we could improve the ease with which lignin can be removed from the plant material, for example. So fixing this so-called recalcitrance problem. And also to try and improve the value of lignin itself, and I'll show you some examples of that in a second. So not so long ago we had this new, it's not such a new concept, but the idea that we, in principle, could design lignins, that we could maybe do a little better than nature herself and design them in such a way that they were more useful for us. And so this is a little bit highfalutin in a way, and yet it's a concept that now is becoming accepted. If you do think about designing lignins, what would you design? Well, one, it'd be nice to have lignins that are easier to remove from biomass, but zip lignins in a little bit. It would be perhaps nice to have homogeneous lignins. Lignins are this big, messy complex, but there are ways to make the polymer with a single structural unit in it. And that makes it valuable both for the polymer itself, but also because in principle you can degrade it to a single product, and that product, the monomer product, might be. And then the other possibility, as I mentioned, you can put other phenolics into the polymer, and so we've found from nature now, where nature's putting very valuable components into the lignin, and this makes those lignins more valuable and makes these products available by the ton in waste streams. And so this is becoming exciting. Didn't know I had this slide still here, sorry. I'm going to highlight the recent progress and some thoughts about lignin products throughout, because I think people are more interested in that. All right. Yeah. Okay, not that crazy. I just wanted to point out that there are a lot of things that have happened in this field lately, and we've been lucky enough to be involved in a bunch of them and to get a whole bunch of covers, honestly, by doing collaborations with lots of wonderful people. But just highlight a few of these things. The first is that this is a pathway that was assumed to be completely known 10 or 15 years ago, and yet we keep finding new genes on the pathway. Don't think I can talk about that, but we'll see. Compositionally, we can change the composition, these H, G, and S units, unbelievably widely by doing very simple manipulations. We can put pathway intermediates now into the polymer, so we don't let the monomers form properly. and yet the plants will still make lignins using the intermediates along that pathway. Some of those are really valuable. I'll show you a couple of examples of that. I mentioned these really valuable components in the lignin. We'll talk about those in a second. And then conjugates. But this zip lignin I'll spend a little bit of time on because this is a rather cool idea, honestly. All right. I was going to tell you about a new gene, a new enzyme on the pathway that managed to get on the cover of Science. That should be worthwhile enough to talk about, even though it's a crappy journal. But, unfortunately, I'm going to run out of time, so and now it's even worse. So I'm not even going to talk about that. Sorry, it's an important enzyme with some nice potential exploitation available. Won't even talk about it. CSE. We'll talk about one because I want to mention it in another context in a second. This is an enzyme. This is an enzyme that puts paracamarate on grass lignins, and we were studying it at the time trying to figure out where this thing is on the lignin, because where it's attached tells you how it got there. And we were able to run some funny old experiments way, way back that were really quite classic in their day, a long time ago, sorry about this, but proving that this thing, this paracamarate, was always on this gamma position of the lignin side chain, never on the alpha position where it was thought to be at one point. And the cool thing about that is it implicates this kind of chemistry. That you take your monolignol, you take your activated paracamarate, and you first make this monolignol paracamarate conjugate, and then you lignify that thing. So this piece lignifies, and you have this paracamarate sort of hanging off the edge. In other words, what's happening is you're taking the monolignals down here, you're taking an intermediate on the pathway, and you're pre-making these little conjugates, and you're sending these out to the wall. You'll see why that's important in a second. This is one of those great experiments where you can not only get the gene and the enzyme, not only get the enzyme and prove their activity, but you can check the gene activity and check the plants that are up and down regulated in this component. So, for example, this is a corn, I'm assuming, let's see where it says that, that has paracamarate on the lignin. So here's the S and the G units, the G's in blue, S and G, little bits of H, and this paracamarate is on it. Very easy to see that. You have a mutant that has that gene missing, and it makes none of those paracamarates. You down-regulate that gene, and it makes a plant with very tiny, tiny levels below what you can detect there. And you can then, of course, up-regulate that same gene and make plants with way more paracamarate on it. So you're sort of establishing the fact that, yeah, you have the right gene that has the right activity that does those things in the plant. And of course, you can look at that in various places. Curtis Wilkerson, he's here, was involved in work on this as well. All right, I mentioned compositional changes. This is, the first part of this is not super new work, but this is very interesting that you can do this kind of massive manipulation of the composition of the polymer without affecting the plant that much. So let me tell you about these high syringeal lignins first. Again, that's not a dog. This is the pathway to coniferal alcohol and synapyl alcohol. There's one hydroxylase gene on the pathway, or an enzyme, sorry, that goes from this guaiacil thing towards this syringeal. By adding the methoxyl, it's the one that puts the OH on, then gets methylated, and makes this synapyl alcohol. Just manipulating that gene can change this G to S composition in the polymer, and it changes it quite spectacularly. So, in poplar, for example, the wild type is 2:1 S to G. You crank down that gene, you crank down that enzyme, you crank down the gene, and you make 100% G, no S. The amazing thing is you crank up that enzyme, and you can make almost 100% S lignin. Why is that surprising? Because nature has done nothing even close to this. This was a surprise, that you would get almost 100% S. These are in plants, by the way, that are indistinguishable from the wild type in the greenhouse and in the field now. So these plants do not care that their lignin composition is changed from 100 to 0 to 100 in a single generation without the benefit of evolution, right? And these plants really are that high. These are 98% S by NMR. You can find the S peaks, you can hardly see the G peaks. They're extremely high percent S. Why do we care? Okay, first of all, sorry, I forgot this was coming. Has nature done this anywhere? Nature's always a good Yeah, always good to look at what nature is doing. She's usually smarter than we are. Maybe not. Canaf's about 7 to 1 S to G. Madrone, I don't know what madrone is, but some tree that's also about 7 to 1 S to G. These are the highest that we found in nature. But our Arabidopsis and poplar, give or take 50 to 1 S to G. So, do we want to do this any better than nature? What are we trying to do here? Well, it turns out that syringeal lignins have higher beta ether content, higher cleavable bonds in them. So if you do pulping, for example, here's what happens to wild type poplar. Here's this high S poplar. You delignify to a greater extent, much more readily, because it has a much more easily cleavable lignin. So I think you can imagine that a paper from this plant requires less bleaching, less delignification during pulping. One of the advantages. The other advantage is something like this. You take the polymer and you decide, okay, I'm going to cleave every one of these beta ether units and I'm going to make monomers from it. And by hydrogenolysis, for example, you can make this set of monomers. The problem is you get this whole set, these H, D, and S monomers. You, unfortunately, if you're not careful, you get some side chain truncation as well. So you get this set of 9 products. But I think you can imagine if you have an S-only product, you only get S products. So now you're down to 3, and if you're a little bit careful, you don't have to get that one. And if you're even more careful with your catalyst design, you can get, for example, just 1 component. So getting 1 monomer component in high yield from a lignin was considered to be inconceivable before. It's now possible with plant materials like this. And it works. So, also a nice paper in Science. Apparently we like that journal after all. It really works. So, for example, beech, I don't know why we didn't use the control poplar, but it doesn't matter, it's about the same. You typically get 40-something percent monomers, and with this high S thing, you get 78, sorry, the graph's tipped, it is 78% monomers. And more importantly than that, these are G and S monomers, so they're already a mixture of at least those 2, and this one's S only, so you have a very simple set, a simple array of monomers that is potentially useful in commercializing. Okay. Yeah, whatever that is. So this is part of the pathway that does this hydroxylation. Where the heck is it? This hydroxylation that gets on the way from this one. Gets on the way from G units towards S units. There's another hydroxylation that's also a little bit interesting, and it takes from these H units towards G units. This is going to get methylated as well. So there's another hydroxylase there, a C3H, that puts this 3-hydroxyl on. That's an interesting one because initially when that was discovered, this is a way to make high H lignins, and it does, but that's the plant. It's not a very happy plant. It doesn't mature, it doesn't get seeds, whatever. It's very tempting to conclude that H lignin is the problem, that this plant cannot deal with having lignin that's made only from this H component. So it sounds like this is a really bad strategy. Turns out that's not true. You can knock out 2 other genes that have nothing to do with this pathway and almost completely resurrect that plant, including to have seeds. And yet this plant has exactly the same lignin characteristics as this plant. I'll show you that by NMR. Always do things by NMR because NMR tells you everything. I don't know where that one went. So basically the wild type plant, S, G, some little bits of H units. You knock out those 2 other genes and nothing happens, but you take that C3H line, the line that can't do the hydroxylation, and obviously that lignin is very, very different. It's nothing like this lignin, but this is the one that's reconstituted by knocking out these 2 other mediated genes, so-called, and then you can make a synthetic H lignin, and it looks rather similar, as you can tell. So this is a plant that has basically 100% H lignin and is doing not quite perfectly well, but almost resurrected in terms of its agronomic capabilities. So the point is there, if we want pure H lignins, we decide, okay, we want these lignins, we want plants with that, and there may be ways that the H is not inherently bad for the plant. And the reason you might want that again is because now you have, in principle, a simple set of monomers that have lower oxygenation, so the fuel value of these is better and they're better certain processes. All right. I mentioned pathway intermediates. Won't get into these, unfortunately. These aldehydes are great. I'd love to tell you that story. They make very interesting plant materials, and aldehydes are really nice because you can do chemical derivatizations and all kinds of things on them, and run out of time. Okay. Talk about these monomers from beyond the pathway. There's a couple that are quite interesting. These were only discovered a couple of years ago. So the first phenolic from beyond the pathway that was discovered is this tricin. Tricin's a flavone, it's a flavonoid pathway, and it's phenolic. It looks a bit like a lignin monomer. Turns out this can only do radical coupling at one end, and it does. So the lignin monomer couples to that end, and you start the polymerization off with this tricin, and off it goes and makes a polymer. So what? Where am I getting to? Okay, sorry, not so what yet. So very easy to see in maize. When we first saw these things, by the way, we didn't know what the heck these peaks were. We thought we had some artifact in there, but turns out it was in old grasses. Very easy to see these tricin peaks. And you can do phylogeny of them, basically, find out where it is that these tricin occurs. They occur on all these camelidid monocots, the grasses, basically. A few other plant materials, really high in oat, wheat, and brachypodium, so quite high levels. Only about 3% of the lignins still. But here's the point again now. This thing is enriched in the waste streams of all these grass lignins, so including corn. I didn't put it on here. I took it off this plant, off this slide, because it got people too excited. This stuff you can clip off by the ton costs $1.2 million a kilo right now. Someone should start a business. If you don't, I'm going to have to. $1.2 million a kilo. I know it won't be worth that when you start clipping it off by the ton. I get that, but it's still very valuable. Right now, it's only used as medicinal because it's so expensive, but it's got all kinds of other potential uses in agriculture, nutraceuticals, and so on. So I predict there will be a great industry based on that pretty soon. And right now, like I say, it's a waste product that's just thrown away over tragic. All right, now I'm going to get silly. Why? What's nature telling us here? So monocots evolved 130 million years ago. Hope people believe in evolution here. Hope so. Primates evolved, whatever. The Australopithecus is about 3.5 to 5 million years ago. Did nature decide, let's put a valuable medicinal on some plants for the evolving primates. And let's put a whole slug of it on this stupid lignin polymer and see how long it takes them to find it. Well, we only found it a couple of years ago, so did not know it was sitting on the lignin polymer in vast amounts. So ready for us to capture. Won't go into this, but basically because there's a gene pathway known for this, it's very easy to knock out tricin, and you can make these plant materials that are lighter in color because they don't have these flavonoid components, whatever they're called, anthocyanins. And you can see that they are knocked out in the stem and in the leaves of those plant materials. They disappear from the animals. Okay. What's that about? Check. The latest thing to be found, not quite the latest, these hydroxystillbeans are being found now. What the heck are hydroxystillbeans? Well, there's one of them you've heard of. What do I have on the next slide? Okay. One of them you've heard of. It's on the next slide. Sorry. These things are in the shells of various palm fruits, like coconut and this macawa fruit. They're in the lignin again. You can release them by the so-called DFRC method, but a reductive cleavage method, and you release several of these things. Pycethanol, there's lots of this thing. There's a thing you can't pronounce, number 2. But there's also this resveratrol. Resveratrol you've probably heard of. It's the component in red wine that's supposed to be good for your health and all the rest. It's in lignin. What? It was only discovered a year or so ago. Amazing. There. Not going to explain this, but the point is that you can find peaks for these things coupling into the polymer by NMR, and you can assign all those dang things if you do enough model experiments. And the implications are that there's more monomers, more pathways to exploit, adds potential value to the lignin, And you can also find, because of, only chemists will care about this, because you can draw resonance forms with the radical from here going all the way up into the top ring, you can find things like this beta 810 coupling up into the top ring. Yeah, only weird chemists will care about that, but all of these products you can find because of the way this just does radical coupling chemistry in a very predictable way. All right. Yeah, and there's one more thing we just found recently. God knows what this is doing, but there's some nitrogenous compounds also found in lignin now. There's one previously found a long time ago, but this guy was just found as well in the coats of corn material. So in principle, a way to get nitrogen into the lignin polymer for time-release fertilizer or whatever is potential applications there. Okay. You're all too young here to know that there was a raging controversy some years back. This idea of engineering new monomers into lignin was once considered to be absolute heresy. And boy, did we get slammed for it in the literature, with statements like this: there is no known precedent for the free interchange of monomeric units in any biopolymer assembly, then or now. Okay. And we went further. Such hypotheses, if ever widely accepted, would drastically change current perceptions of how and strategies for achieving numerous biotechnological goals. This was supposed to be damning to us. This was a criticism of how stupid we were suggesting such things. It turned out to be unbelievably prophetic, because this is now what everyone's doing. You can put these additional components into the polymer, you can make use of them, and you can get extra value out of them, and everyone, a lot of people, are working on changing lignin this way. So that thing that was supposed to be very denigrating towards us and all the rest turned out to be quite prophetic, and I can't help but smile. All right. Conjugates. Fortunately, we're not talking about those. There are a whole bunch of conjugates that have little components that you can clip off and get value from them off the polymer. I won't talk about those, but I will talk about this one case of zip lignins. This is a very cool idea, and I can say that because it didn't come from me directly. It came out of a little group dynamic, and we can't actually figure out who had the exact idea at the exact time. It seems like we all had it at sort of the same time. It just came out of a whole bunch of studies that I'd love to go into at some point. But this is the idea that an idea that is 15 years or more old now, that we can redesign lignin for our purposes and maybe teach nature to do something new, something we hadn't thought of. That's the idea anyway at the time. The thought behind it is sort of this. You take some poplar lignin, and the weakest bonds in the polymer are these beta ethers, but those weakest bonds still require really stringent conditions to cleave them. So you cleave the bonds in the polymer to cleave it to smaller molecular weight components to get it out of the way of the polysaccharides, for example. But 170 degrees in caustic soda, a couple hundred degrees in acid, that's the weakest bonds. Wouldn't it be great if we could put more easily cleavable bonds into the backbone of that polymer so it would fall apart more easily? That's the idea. Wouldn't it be nice if we could put a few twist ties in so that we could just untie the structure much more easily? And so the idea is either insanely clever or really stupid, depending on where you're sitting at the time looking at it. But the idea was to take these monomers again, coniferyl alcohol, synethyl alcohol. Remember that we coupled it already to paracamarbyl-CoA to make these monolignol paracamerics that are in grasses. But what we're going to do is take frulyl-CoA and make these monolignol conjugates. What's the difference? They look a bit silly. They look like dimers. But we realized there's two features of these things. And the first is that these ferulates radically couple the same way as the monolignol does. So these couple at both ends of this sort of dimery thing, this conjugate. Can't draw, sorry. But you end up making, you have a component that you incorporate integrally into the polymer itself at both ends. But what you realize you've done when you do that is you've integrated a simple little ester bond right into the backbone of the polymer. And it's very easy to clean esters. First year organic chemistry, you know how to do that. So the whole idea is if you make the lignin this way with these things incorporated, it should fall apart even if you temper it. Good idea. The reason it does that is because it's got these esters, not just near the end of the polymer, but these esters incorporated into the very backbone of the polymer. All right, I won't tell you how we found the genes and all the rest for that, but did get a gene out of Chinese Angelica, popped it into poplar, devised a method that I also won't go into for proving not just that you had these components made, but that they went into the polymer and made and got incorporated into the polymer. And then you could determine that in the transgenics you release this coniferal ferulate and synapyl ferulate, for example, from the polymer. Got that into a modestly decent journal again, didn't get the cover, that's always annoying. In that, sorry for people who have seen this, in that paper we said these are perhaps the first plants that are designed to be deconstructed more easily, designed for deconstruction. So we can write that. What we can't do is write the news items that come out about this paper. So what was written in the news headline was cheaper fuels from self-destructing trees. The internet lit up for a few weeks with people complaining that they'd be walking in the forest and these self-destructing trees would be exploding all around them. So, please let me assure you, these don't self-destruct in nature. They're chemically easier to cleave apart. They're not self-destructing by any means. All right. These work rather well in the field. We've got them growing now for a few years, and I'll only show you one result from pulping. With pulping, you're trying to delignify, get down to a certain lignin level that you measure by this kappa number. I won't explain what that is. Kappa number, and in the wild type, under the same conditions that you delignify a wild type 2, you get much higher yield from the transgenic because it delignifies easier and then you protect your carbohydrates more under those conditions. You can do some quick economic analysis, and it's worth quite a bit to the mill and to the industry in general. That's for a very low level of ferulates, these conjugates incorporated into them. And there's considerable upside yet, so we're trying to increase the level of those things. All right. So it seems like we have perhaps successfully designed these plants for deconstruction, and the energy required for processing is lower indeed. So, aren't we clever? Let's see you try and beat that nature. Nature's pretty clever. She does all kinds of things that you might not expect. If any of you saw this in this slide that flashed past before, come work in our lab. We want you. Look at this. What? So here's where I said we made these conjugates. But look at the wild type. What? These peaks are in the wild type material. These conjugates, they're already being made in the plant at low levels. And it turns out you can measure the levels in a whole bunch of plant materials. It's in all the grasses. They make mostly synapyl ferulate, and about half the hardwoods make coniferal ferulate. And I'm not saying this is the reason, but all these hardwoods that have this CYP gene in are the ones we've chosen for pulping. They pulp the easiest. Again, I'm not saying that's the reason, but I think we've already been selecting for these guys. So, just interesting. So Mother Nature already invented that before us. Dang annoying, but actually it's quite nice because it means we're not putting something new into the environment. But, all right. I'm seriously worried now. We got off to a late start. I have about, 10 minutes left, but I'm worried that we're going to go over time. What are we doing? I'll just go? All right. Yeah? Okay. You guys can stand it. I think I can. All right. I want to talk about lignin utilization just a little bit, and there's one cool thing in here that hopefully is worth getting to. I'm glad people here are talking about utilizing the polymer. We can't work on that. Polymer is way too difficult for us. We have no idea what you do with the polymer, so please go do that. It's very important. We, because we're chemists, we can produce monomers from the polymer, and these monomers, phenolic monomers, are potentially really valuable. And recall again that lignin is this largest source of aromatics on the Earth, sustainable aromatics. Only 3 ways really of making monomers. It's not quite true, but you can hydrolyze the lignin, you can oxidize the lignin, and you can do hydrogenalysis of the lignin. I'll show you just a couple of those, but what's important about it. But here's the problem. Lignin has a nasty feature in that it condenses with itself, not that kind of condensation. The problem is this. It's very easy to take these benzylic alcohols, you throw them in acid, for example, this thing pops off and you make a carbonyl. Some of you remember enough of your chemistry to know those are a little bit reactive. And the trouble is that not only do you have this carbonyl, but all the rest of the rings in the lignin are quite electron rich. And so what happens is these things couple with each other, that's on the next slide, these things couple with each other and you make brand new bonds in the polymer. And good luck ever dealing with that bond again. You're never going to cleave that guy again. So this condensation is a problem when you try to get the lignin out, particularly with acid, for example. So the problem is the monomer yields are usually quite low, 7 to 26% or whatever it is. The theoretical yields are around 40 to 50%. Here's why it's so bad. And we were talking about earlier about this too. If you do any kind of acid isolation of your polymer, so for example, this is organosolv, this is ethanolysis so-called. You get very nice lignin. In this case, it's a mild condition. Here's your S and your G components. It's got some para-hydroxybenzoate because it's willow, it doesn't matter. And in the side chain, you see these beta ethers, the blue things, and then these other structures you recognize too, the green stuff and the purple stuff. Very nice lignin that looks like a native lignin that we would isolate laboriously from the cell wall. If you just up the severity of the conditions, you get something like this. And you're going, okay, that's that, and that's this. And the problem with this thing is, I don't recognize anything except that stupid methoxyl group up there. So you've totally rigged this lignin, and yet both of these things are called the same thing, and they might be sold as the same thing. So be very careful when you order these things. These are very different. The mild one looks like native lignin. Our lignin knowledge works. We understand the chemistry of that a fair bit. The harsher one looks like junk. It doesn't mean that it's completely useless, but it is useless to us if we're trying to make monomers from it by cleaving beta ethers, for example. There are no beta ethers in here. So this is a big mess, and you could easily get stuck with a polymer like this that you thought was one like this, and you will never be able to do anything good with it in that sense. There is a nice way to get around this condensation problem. The first way, Jeremy Luderbach came up with it, is to add formaldehyde. There's a better way now. But you protect these 1,3-diols so you don't have an OH here anymore. You actually end up derivatizing the ring a little bit too. But when you do that, you stop that condensation reaction and your monomer yields now go much, much higher. And that works. So that was that paper in Science again that gave us the 78% yield. All right. At about this point, you start to think about how would you design the polymer if you wanted an ideal lignin for a biorefinery? What kind of properties would it have? And Yanbing Li in my lab did a good job of thinking about this process. Wouldn't it be nice if it was acid resistant, so it didn't have this condensation problem? Asking a lot. Wouldn't it be nice if it only had CO bonds between units? Because in principle then you could cleave every single one of them. Wouldn't that be nice? Wouldn't it be nice if you only had one type of unit so that you only get one type of product? Well, you might have already provided an example again. What the heck is that? So this is vanilla. And if you look at the stem of vanilla and you do bioastalysis or something, it has SG lignin, normal old SG lignin. I won't explain what that's about, but nice analytical technique for determining the lignin composition. If you look at the seed coats though, it has something that's not either S or G. It turns out to be C. It turns out to be designed from cafe alcohol. Only one component. It also turns out, unexpectedly, that this thing makes a totally regular homopolymer. This makes it really valuable for making carbon fiber and all kinds of things from that normal lignins aren't, because normal lignins are a big fat mess. It also means, where am I going with that? So, it also means it's only got one type of subunit. It contains only CO bonds. They're slightly different ones because it made this ring structure, but they still are only CO. In principle, they can be cleaved. And it could be acid resistant because you don't have that benzylic OH there anymore to make those carbonyl bonds. Turns out that's actually right. If you take a normal lignin and you throw it, so this is the S and G again, and the beta ethers, for example. You throw this in conch sulfuric acid, and you destroy it. Don't recognize anything. You throw in lithium bromide, also acidic. You keep one component, but all your beta ethers are gone. You've destroyed it again. You take this C lignin, this lignin from caffeal alcohol. It really only has this alpha, beta, and the gamma peaks. Everything else belongs to carbohydrates or whatever in the thing. Very simple spectrum, just those 3 peaks. You throw in conch sulfuric acid, they're still there. It's stable. You throw it in lithium bromide, they're still there again. No matter how harsh your acidic conditions are, this thing just sits there like a brick. This is a great way to isolate a polymer in a fairly pure form without changing it and without breaking it. And if you do hydrogenolysis, you get 2 monomers from it very, very nicely. You do it from the polymer, you get 2 monomers plus another one, and all that other rubbish is from the other components in the wall. And that's just about done. If you do this hydrogenolysis on, for example, spruce, you get a lousy 20, whatever that percent yield is. Birch, you get a bit more yield. The highest poplar, we were getting 78%. Again, I'm projecting the stupid 3D plot, sorry. And the C lignin one, we're getting close to 90% yields of just these 2+ monomers. Once again, though, these were G only. This is a mixture of G and S. This was S only. This is C only. So you're making just 1 monomer. And you can choose the catalyst somewhere. You can choose the catalyst to strongly favor one. So we're getting a 90% yield, 90% of which is 1 single monomer. I think the idea of getting a single monomer and 80% yield from a lignin is just, again, inconceivable not very long ago. All right. So you can imagine that we're all trying to, you can imagine that we're all trying to make these plant materials with these intermediates now, these caffeal alcohol and 5-hydroxychemical alcohol. So there's a renewed emphasis on trying to make this a real plant material, make this available by the time of year. All right, conclusions. We can design lignins for improved deconstruction. This is the lignin idea, that sort of works. To make biomass that's easier to pre-treat. We can make novel and even homogeneous lignins. Nature's shown us the way there. We didn't invent that. Better and simpler products, there's high-S ones and so on. And we can incorporate valuable co-products, things like this tricin and resveratrol into the lignin, making them available in a waste stream by the ton for our exploitation is a word I hate to use. All right. Don't know what I was going to say here. Sorry. Nature's damn clever. Okay. Given a few billion years, all kinds of things are possible. And we should probably not get too smug about what we can do. Again, we thought we were clever a couple of times. Yeah, nature showed us up every time. She's much more clever than we are. We should have known. But nature is really good at inspiring us, so good to look towards nature for inspiration for the kinds of things we're trying to do. All right, lots of people thank you for your patience and listening. Thanks.
I think you have 10 minutes for questions, for a couple of questions, and then we can go to the next room for receptions. Goodbye.
Students have to ask questions. No, not you.
John, if you can incorporate resveratrol, can you make it in the plant and store it without having to incorporate it first?
In principle, right? You particularly, so the question was, can you make something like resveratrol in the plant and not incorporate it into the polymer? I sort of like the idea of incorporating into the polymer just because you have a pull on it basically, and you have a way of isolating it by the ton that way. But there is a way. Basically, plants will detoxify materials by glucosylating them and sticking them into vacuoles. And so you can make decent quantities of components like that and store them in the vacuole. Yeah, if they don't go to the wall, I'm not sure how you stop them getting to the wall apart from glucosylating. But in principle, yes.
So if you've got intermediates, aldehydes, as well as hydroxyls, what about acetal linkages for unzipping?
Ooh, that's a good question. Yes, acetals are great.
I just wondered
So-
if there was something in
Yes.
nature that, again, might lead you to down
It
that
turns
pathway.
out, so acetals on carbohydrates, I don't know if we've figured out a good way to turn the aldehydes into things where they've got two sort of phenolic ends that you've connected together. That actually hasn't been done. But one way is you can incorporate ferulic acid, not at very high levels, but ferulic acid itself into the lignin. And it turns out one of them decarboxylates and it makes an acetal by double beta 4 coupling, doesn't it? Makes acetals, and that material is indeed more amenable to simple free treatment in acid that falls apart more easily. So-
Yeah, I think
so-
there are some lipid acetals from
yes, exactly.
exactly. That's why I'm
Yep.
bringing this
Yep.
up very much
So,
so.
actually,
As well as
so-
polyacetal as a low polymer.
So you should patent that right now. No, because I don't think anyone's thought about taking the aldehydes and making some kind of diphenolic acetal. Yeah, that's a very good idea. I can patent that tomorrow.
Just put me on the path.
Good idea.
It's too late. It's in it. It's
Patents are worth so
Yeah.
much.
patents. Not.
Yeah, go.
Me?
Yeah.
Okay. You said a little bit about this, the chemical coupling process of lignin polymerization. There is something, okay, so all the monomers are produced from the cell, and then there is some kind of process that involves enzymes to make this as radicals.
Absolutely.
Do we know much about this kind of process,
Yes.
where it happens and how?
So, yes, in principle. So Ding's right. So I said the polymerization process is purely chemical. It's absolutely true. But all the way up to making the monomers is absolutely controlled by the cell and all the enzyme machinery and all the rest. And making the radical is actually a lactase or peroxidase process. So enzymes are required up until that point. It's the polymerization, the polymerization to the polymer itself that is independent of control. That's still important, but you're right. So it turns out lactases were thought to be not important, only peroxidases were important in lignification for a while. Turns out you knock out very specific lactases and it affects lignification like you couldn't imagine. So yes, those things are becoming known and are definitely important. And no, we don't know why. Why one enzymatic procedure to make the radicals is favored over others in certain tissues or whatever. Totally unknown as far as I know. Yeah. So
Okay, so
I don't know.
you have actually you are really making a new definition of lignin.
Sorry.
So the new definition of lignin.
Okay.
What is lignin? So
Ah.
so now we usually thinking about the lignin has three different kind of monomers, let's say HSG. Now you have new components in lignin polymers. So can you, maybe
No,
it's
so-
a good time to give a good definition about what is lignin now.
But so we've had some real flack about that for good reason. Again, lignin was a very simple definition. You had to start with one of those three monolignals and you made the polymer of that. And there are certain people that say that is the way lignin is. Turns out there's no such thing in nature. Nature couldn't care less about your stupid definition, right? So it'll put in whatever it decides to. So the definition has to include anything that gets sent to the wall as a phenolic that can end up in the polymer. I mean, sorry, it just has to. So we submitted a paper one time with, what was the okay, I'm going to say it was the Café El Alcohol one, but I'm not sure that's true. It doesn't matter. And we said this is a pseudo-lignin or lignin-like component or something like that. And one astute reviewer actually said, oh, for goodness sakes, just call it lignin. It's undergoing radical coupling the same way, the same mechanism. You get products that are not optically active, so it's still a chemical process. You know, the plant's going to call it lignin. It probably is. So the debate is still on, and I don't know where it's going to come down, and I'm not sure I really care, but for sure, the plant will make a polymer that's useful to us by whatever it has at hand, basically. And I said this 25 years ago, and I was out hell for it. The plant couldn't care less about the exact structure of the polymer. I still totally believe that because, it's showing us that all the time. The exact structure couldn't care less. it needs certain properties to function for strength and water conduction and all that kind of stuff. Not interested in controlling the exact sequence of units for each. And the monomers too. Obviously, you can flexibly introduce some monomers. Some of them will cause all kinds of problems, right? You can't just take any old thing from the Aldrich handbook and put it into the plant and assume it's going to be fine. But there are plenty of examples where the plant is happy with that component. So, yeah, so I think the definition has to change to be more encompassing. Right? Yes. Any students? Students have asked questions. So shy.
They're probably gonna ask questions
Okay,
in the other room.
yeah. Sure. Don't be shy.
There's no more questions. We let John, we do his time, we'll go out again.